Rhizopus species. Rhizopus, or commonly known as the bread mold, because they are found to be growing on the stale bread under high humidity condition. This species of fungi is considered mostly for the negative impact, like the storage and transport disease of grape fruits like mango, peaches, strawberry, and also causing the disease mucormycosis. But apart from this negative economic loss, there is also certain economic importance to it. For example, various species of Rhizopus, they are responsible for production of industrially important products like lactic acid, formic acid, citric acid, as well as oxalic acid. This cottony mass of hypha, fungal hypha, as we are seeing, this cottony mass is actually the second stage of infection. When in the first stage of infection, the hyphal mass grow among the substratum, within the substratum, and only when the feeder mycelium has reached a certain stage of maturity, then only we see these type of surface mycelium. This white cottony mycelium that is present on the surface of strawberry, it is the aerial mycelium that is in the second stage. After the aerial mycelium has it has reached a certain stage of maturity, that is accumulated a high amount of reserve food matter and oil droplets in its cytoplasm, then only the reproductive stage begins. Hello friends, I am Dr. Ayantika, and in this video I will be discussing about reproduction and life cycle of Rhizopus species. In case you want to get an overview of what Rhizopus is, I have made an earlier video where I have discussed an overview of Rhizopus, that is the morphology, anatomy, as well as ecological, economic importance of Rhizopus. And link to that video I have put in the description box. You can go and watch that video in order to have an overview before plunging in the reproduction and life cycle. You can go and watch that video also. So here in this video, I'll be discussing about reproduction and life cycle, which is in the BSc and MSc syllabus of various universities. And so the reproductive phase only starts when the feeder mycelium as well as the aerial mycelium has reached a certain stage of maturity. That is, they have accumulated high amount of lipid droplets in their cytoplasm. Then only this mycelium starts its reproductive stage. So let's begin the video. So the reproduction is of three types, that is vegetative, asexual, and sexual. As we know that Rhizopus, it belongs to the class Zygomycetes, that is, it is a terrestrial fungi. So all the reproductive stages, any of the reproductive stages do not have any motile reproductive stage. Like the zoospores, they are totally absent. And in its absence, we have sporangiospores or chlamydospore. So the reproduction, the vegetatively it reproduces by fragmentation, especially in the early stages when the aerial mycelium, by accidental breakage, this type of fragmentation occur, and each of these fragments by accidental breakage, say for example this is a fragment, and each of this fragment will produce, they are fully capable of producing a new mycelium. So this is the vegetative reproduction. Then we have this asexual reproduction. So asexual reproduction in the form of sporangiospore as well as the chlamydospore. So sporangiospore is produced when there is abundance of food and the temperature and humidity is very conducive for growth. But when the temperature is high and the humidity becomes low, there is drought stress, water stress, then the chlamydospore are produced. And the sexual reproduction occur after several rounds of asexual reproduction. Then only the sexual reproduction occur. That is when the fungal hypha mycelium is fully mature and is loaded with food material. So sexual reproduction, it is occurs by the gametangial conjugation, where the entire content of the gametangia fuses, and this type of gametangia, it is called the aplano gamete, and there is no motile spores, so the aplanogamete is just a mass of sporangia. And I will be discussing all the three type of reproduction in detail in the next few slides. So the sexual reproduction, one thing is that the hypha is two types in most of the species of Rhizopus. The hypha is usually either they look morphologically similar, but genetically or uh, physiologically they are different. So this opposite type of strains, one of the strain is designated as a positive strain, another the strain is designated or labeled as negative strain. So most of the Rhizopus species, they are heterothallic, which means that there will be two strains of hypha, and these opposite strains of hypha, only when they come in contact, they have the sexual reproduction. But Rhizopus sexualis is the only species of Rhizopus which is homothallic. That is, same physiologically same type of hypha can fuse to form the zygospore. So we see just only one, that is one species, Rhizopus sexualis, is homothallic. It is Rhizopus sexualis is homothallic, and all the rest are heterothallic. So the asexual reproduction, it reproduces by either the sporangiospore, chlamydospore, or oidiospore. So the sporangiospore is the most common, and when we know that the mycelium, after it has grown, if this is the substratum and this is the mycelium growing on the among the substratum, so after it reaches a certain stage, the mycelium, it becomes aerial, and stolon, they form stolon, which these stolons, when at the point of they grow horizontally, and at the point of contact with a hard substratum, they the tip bends, and they produces rhizoids. On their lower surface they produces these rhizoids, and on the upper surface of the stolon node they bear this type of sporangiospores. So sporangiospores, basically when the aerial hypha, it gets vertically upward, and the tip of the hypha swells to produce the sporangiospores. So these sporangiospores that is produced inside this sporangia, they are haploid, that is mitotic, and this is a very common method of asexual reproduction. This is the nodal region, and the sporangiospore are produced only in the nodal region. We know that in Rhizopus, in the nodal region from the lower surface, these rhizoids appear, and on the upper surface, these sporangiospores appear. So this is how the sporangia is formed. So any upward the vertical hypha, the tip of it gradually, here in this diagram, the tip gradually swells, and as the tip swells and increases in its size, there is gradual differentiation of the cytoplasm. The peripheral portion, that is this portion, the peripheral portion is becoming densely it has got dense cytoplasm, and is filled with nucleus. And we see that there is several nuclei, these black dots. These are the nucleus. These black dots. These are the these these are the black dots. These black dots. Sorry, this one. These black dots. These are the nuclei. And the central region here in this central region, we are seeing that it is highly vacuolated. So inside, in the tip region, terminal region of the sporangiophore, we are seeing that they have a dense peripheral cytoplasm and vacuolated central region. And the peripheral region, this is the sporogenous region, and this peripheral sporogenous region, it here we see that gradually this region is getting segmented into these multinucleate segments we are seeing, and each of these multinucleate segments, they round off to produce these spores. These are the spores. Here we are seeing that these region are getting they are forming spore. And what is happening in the central region? In the central region, the vacuoles in the periphery of the central region, here these vacuoles, they fuse with one another and thus separate the vacuolated region from the sporiferous region. So this vacuolated region is called the columellate region, and the outer sporogenous region that ultimately form the spore is called the sporiferous region. So this they become separated, and this is how the spores are formed. This is the diagram of the spore. So as the spores become mature, the central columellate region, that is the central columellate region which was present, and this is the outer sporoferous region, so which contains various spores. So these are the spores. Say for example, this is the spores, and I have drawn only one spore, but it contains many spores. So as the spores become very mature, so this central columellate region, they accumulate more and more fluid, and they exert a pressure, and that pressure, it causes a breakage in the wall of the sporangia. So once there is breakage in the wall of the sporangia, the spores get exposed. And once the spores are getting exposed, and this sporangial wall, they dehisce, and they remain as a collar-like region, and the columella, it forms a inverted cup-like structure. Here we are seeing this columella, it forms an inverted cup-like structure, and the spores are getting fully exposed. And these exposed spores, when the spores are mature, they are getting the wall is getting dispersed. After dehiscence of the wall, the all the spores, they are exposed. So a slight breeze or any wind current will cause the dispersal of spores. So Rhizopus, the sporangiospores, that is haploid, they are dispersed by wind. So this is how the spores, they are dispersed, and the sporangiospores. So after the spores are getting dispersed, and if they fall on suitable substratum, that is substratum containing high amount of starchy matter, and if the temperature and the light condition is very conductive, then immediate germination occur. But if the humidity condition or light condition is not good, then it can perinate. Even certain species of Rhizopus has been found to perinate for 10 to 12 years. These sporangiospores are found to perinate and only on return of favorable condition do they germinate. But when they germinate, they absorb water, and as a result of absorption of water, it swells. So the outer wall, that is the outer spore wall, it forms a small ridge, and it burst in that region. And before it burst, germinating layer of that is a cell wall, germinating cell wall layer is formed. So the enclosed in the germinating layer, this entire cytoplasm, it protrudes in the form of a germination tube. So in this way it grows. So we see here the spores, and then the spores they swell up, absorb water, swell up, and the germ tube is formed. And after the germ tube is formed, the germ tube, it elongate, it branches, and gradually this type of mycelium is formed. So this is how the entire how the sporangia is formed. Sporangiospore produced inside the sporangia, which get disseminated by wind, and they fall on suitable substratum, germinate to produce this type of mycelium. Then we have the chlamydospores. The chlamydospores. This is how exactly the chlamydospores look, and this is the electron microscopic form of how the chlamydospores look. So chlamydospores, unlike that of the sporangiospore, which are formed during favorable condition, chlamydospore are mainly the perinating structure. So in absence of or in when the amount of water is low, the entire hyphal mass, what happens, the entire hyphal mass, it gets segmented, it undergoes septation, and each of this septated region, they accumulate lipid bodies, the starch granules and all this oil droplets. So they accumulate the food matter, and they grow a wall. So this is the this thick-walled uh, f- uh, this thick-walled nutrient-rich spore. It is called the chlamydospore, and they each of the chlamydospores, they have intermediate region which is practically dead, and this region, when they desiccate and set free the chlamydospore. So the chlamydospore, they occur intercalary position, and by they are set free by the decay of the intermediate region. Though in Rhizopus stolonifer, which is a model for it is a type of type specimen for Rhizopus, this type of chlamydospore is absent, but or rarely found. But chlamydospore is found in other type of Rhizopus species also. So these are basically these chlamydospores. They are formed under drought condition, and they can perinate for many years until the return of favorable condition. On return of favorable condition, they germinate to produce a normal fungal mycelium. This is the oidiospores. This one. Actually, this oidiospore are produced when the fungi is grown in liquid media or very nutrient-rich media. Then this type of oidiospore are produced, and they look like the Saccharomyces type of yeast. So that is why they are called the Torular stage. And these type of yeast-like bodies, they are formed when the fungal hypha, they under very high nutrient condition, they undergo septation, and each of this septated portion, they separate, and each of this septated portion give rise to a new mycelium. So each of this, they give rise to a new highly branched fungal mycelium. So this is the last type of asexual reproduction. Then we move on to sexual reproduction. where we know that in Rhizopus, most of the species they are heterothallic, that is, they have physiologically two type of mycelium. One we have designated is as positive, another we have designated in as negative. But one species, that is Rhizopus sexualis, it is homothallic, that is, they have only one type of physiological mycelia. So what happens is that in most of the heterothallic species, when the mycelium is growing of mycelium of the opposite strain is growing close enough, then only there is a slight protrusion of the mycelium. And this type of here we see this is the slight protrusion of the mycelium, and this protrusion, it is called the progametangia. This type of protrusion occur only when the opposite strain of mycelium is present. It has been found that there has been certain chemicals that are released only when the opposite strain of mycelium come in close contact. So once the opposite strain of mycelium come in close contact, this type of protuberance called the progametangia is formed towards the opposite strained mycelium. So this type of protuberance, um, what they happen is that here in this diagram we are seeing that this protuberance, the tip of the protuberance, that is the tip of the protuberance, once the protuberance is formed, the tip region of the protuberance, they get opposed to one another, and the tip region, they show swelling, and this swelling is due to accumulation of dense cytoplasm rich in nuclei. And so after this type of swelling is formed, there is a septation, as we see that this is the septation formed in each of the progametangia. And after the septation is formed, this terminal region is the gametangia. So this terminal region is called gametangia, which I am marking in red. And this basal region is the suspensor. So this is also the suspensor. So after the septation is formed, the progametangia is divided into basal region, suspensor, which is much vacuolated, and terminal region, that is the gametangia. Terminal region is the gametangia, and that gametangial region is rich in storage matter, as well as dense in nuclei. So this gametangial region, the membrane that is opposing both the gametangial region, that is this region that I'm drawing in green, this membranous region, it dissolves. And when it dissolves, there is mixing of protoplasm of both the gametangia. And this mixing of protoplasm of gametangia, that is the fusion of the two gametangia. um, this is the sexual reproduction. And after fusion of the gametangia, the content of the gametangia, this structure, that is the zygospore, is formed. And actually, the after fusion, prozygospore is formed, and the prozygospore, only after it forms a wall around, thick wall around it, is converted to zygospore. So the zygospore, after it is formed, it undergoes a period of rest, and then after a period of rest, do the germination occur. So it has been said by certain scientists, it has been postulated by certain scientists that after a period of rest, the content of the zygospore, the content of the zygospore, it undergoes reduction division, that is meiosis, where the diploid zygospore, it is converted to haploid. Diploid zygospore is converted to haploid, that is from 2n to n. But certain other mycologists believe that this type of meiosis, it occurs before the resting period. So this has been a matter of controversy. But however, the after germination, when the germination starts, at the time of germination, there is a haploid condition has been restored. So when the zygospore is mature, then that is after the period of resting. So after the zygospore has matured, that is after the period of resting, the zygospore, the content of the zygospore, they they germinate to form a germ tube. By this time, that is during the period of resting, this suspensor cell, which I am drawing in yellow, these suspensor cells, they have desiccated and they have liberated the zygospore from the mycelium. The mycelium is also dead by this time, because reproduction usually occur when there is a drought stress. So this suspensor cell has dried up and liberated the zygospore. So after that, the zygospore, they germinate. And this germination, it occurs by the formation of a germ tube, which I'm going to discuss in the next slide. That is here, this is the picture. So the outer layer, as we know that the zygospore was formed of after from the prozygospore, only after it has laid a thick wall, zygospore was formed. And that this thick wall, which consists of outer exine and inner, this region that I'm drawing in yellow, this is the intine. So these electron dense exine and electron transparent intine, they help in tiding over the period of drought. So on return of favorable condition, what it does is that the zygospore absorbs water, it swells up, and there is a rupture of the exine. And the content of the zygospore in the form of a promycelium, germ tube or promycelium, comes out, and then it rebranches to form a vegetative hypha. This is the vegetative hypha we are seeing. This is the vegetative hypha. And the tip of the vegetative hypha, they form a germ sporangium. That is the tip of the hypha. This type of sporangium that is born on the tip of this hypha is a germ sporangium. And that germ sporangium, inside it contains germ spores. Now these spores are meiospores because they are formed after meiosis. That is the zygospore that is formed after the sexual reproduction, that is gametangial conjugation of the two aplanogametes. Uh, this after period of resting, this zygospore undergoes meiosis, that is from the diploid condition, haploid condition is obtained, and these are the meiospores, that is these haploid spores are called the meiospore, that are present where, that are present inside this germ sporangium. So germ sporangium contains the meiospore, which liberated and produces the mycelium. But there are also other type of alternative things, that is in case there is failure of the gametangial copulation, that is this stage, this is the failure of gametangial copulation in case, then what happens, then the zygospore is formed parthenogenetically. That is the gametangia that we know that if this mycelium, that is a progametangia, they do not fuse, then each of these gametangia, they develop parthenogenetically into another spore like structure. But this spore like structure, it is haploid, and this is called the azygospore or parthenospore. So this azygospore or parthenospore, this azygospore, which I'm writing as A, they on germination, the spore on germination as zygospore, they produce a haploid mycelium just like that of the normal sexual reproduction, but the middle stages, that is fusion, formation of diploid zygospore which germinate and produce haploid germ sporangium, these intermediate stages are missing. So we see that in absence of sexual reproduction, this azygospore is also produced. Now about the life cycle, we are seeing that when there is and this is the mycelium, we begin with this, the rhizoidal hypha. After a period of growth, they undergoes vegetative asexual reproduction, that is the sporangiospore. These are the vertical sporangiophore. The sporangiophore are produced, the tip of which swells up to produce the sporangium. That is, these are the sporiferous region, outer peripheral sporiferous region, which produces the spores. And this each of these spores on liberation, again the spores on liberation again repeats this mycelial stage. But when the mycelium is old enough and has accumulated the food reserve, then this type of sexual reproduction occur, where opposite strain, we are seeing that the negative strain and positive strain, opposite strain mycelia, when they come close in contact, they develop a progametangia, and the tip of these progametangia, they undergo swelling, and then the tip region, that is this tip region, which is called the gametangia, they they undergo fusion, and as a result of this fusion, this zygospore is produced. And this zygospore, actually the pre-zygospore is produced, which undergoes on formation of the cell wall, and after formation of a thick wall, it is converted to zygospore, which undergoes a period of resting. And after a period of resting, this zygospore, they germinate to produce a mycelium, which produces again a germ sporangium at its tip. The germ sporangium, it produces inside the germ sporangium are the germ spore or the meiospore, which produces a fungal hypha. Each of these spores produces a fungal hypha. Now, one thing is that the diploid phase, it starts with the zygospore. That is, this is the diploid phase, the 2N phase, the 2 the 2N phase starts here, and with the formation, with the germination of the zygospore, this the diploid stage is in this stage, the diploid stage is lost. So only in these two stages, that is in this two, that is the zygospore stages, only the zygospore stage is the diploid stage in the life cycle of Rhizopus. So that was all about the reproduction and life cycle. And I do make videos on other botany topics like algae I have made, and cyanobacteria, lichens, fungi, pteridophyte, bryophyte, and I have put all those in the in a separate playlist. You can go to my channel and watch those videos. If you like this video, do hit the like button, share it with your friends, and also please do subscribe to my channel, because your subscription, it encourages me to make more such videos. And if you have any doubts or queries regarding this or any other video, you are free to write it in the comment section. Also, if you want me to make videos on any topic, do write it in the comment section. Thanks again for watching this video.